Hello, everyone. Welcome to Backyard Musings, broadcasting live from Apple Valley. I'm Steve.
I'm Scott.
Good morning, and thank you for joining us. A recent study conducted by Lund University in Sweden has resolved a long-standing mystery, identifying which patients with aggressive breast cancer benefit from a targeted cancer treatment that was previously under development but had been discontinued. This breakthrough is significant, offering renewed hope that the drug's development can resume and ultimately reach the patients who need it most. Would you like to know more?
So, despite extensive research and development by North American scientists spanning many years and costing millions of dollars, the drug ganitumab, which I don't know who came up with that one,
I know.
has shown promising results against breast cancer in animal trials, was ultimately shelved. Well, I don't know why. While it underwent clinical testing and achieved some success, the critical question of which breast cancer patients would benefit from the treatment remained unresolved. When developing new cancer therapies, it is often the case that not all patients with the same disease are helped by the same treatment. That is why clear diagnostic markers are needed. It was not possible, however, to find clear indicators that could show healthcare professionals which individuals actually benefit from the treatment. The medicine blocks a receptor, IGFIR, on the tumor, which then activated can lead to the tumor growing and spreading.
Yeah, interesting. Very interesting.
Yeah.
Quote, it was a great disappointment to many of us in the field. We saw clear results in pre-clinical trials, but when we looked at entire patient cohorts, it was not possible to draw firm conclusions. So when I saw the results from the research group in Sweden, whose analysis came up with the answer as to which marker identified those who benefited from the drug, I was very pleasantly surprised, says Michael Pollack, professor at McGill University in Canada, and one of the co-authors of the study. Fortunately, the research in the USA have made all their data from the I-SPY 2 trial available. Perhaps other researchers will be able to solve the problem. Again, sharing of information. Let's share it. My goodness.
Yeah. Why don't they do that? I mean, it's almost like when these guys are doing this, it's like they're inventing something, which I get that if it's going to be
proprietary. I get it.
Yeah, but I mean, if you're trying to save people's lives, I mean,
but it's data of who benefited from what and what indicator or whatever they called it, the identifier. Share that information.
Exactly. I mean, why go down? I mean, again, I don't know that deep into this thing, but if you're doing something and it's working, why wouldn't you want to share that with all the other guys that are trying to do the same thing? Anyway, so Christopher Godina, a postdoctoral researcher at Lund University and a junior doctor at, is that Skane University Hospital?
I think so, yeah.
Was drawn to all the data collected after the clinical trials in Sweden and Canada. In the clinical studies, patients had been given the new drug before surgical treatment for breast cancer. But Christopher Godina looked at the data from a new perspective. He mapped out the way in which various genes are expressed in the tumors and the ongoing processes around the cancerous tumor and compared this with patient outcomes. This involves advanced bioinformatics calculations, combining biology, computer science, and mathematics to analyze biological data. Focusing here on the genetic, molecular, and clinical data. The aim was to see if it was possible to find any marker that the patients who were helped by the treatment had in common. Surprisingly, it did not matter whether or not the tumor had the receptor the medicine was supposed to block on the cell surface.
That is a little bit surprising. Okay, here's a quote. We found a biomarker, IGF-BP7, that helps us to show which patients may benefit from this treatment. In the group with lowest levels of the marker, almost 50%, in 15 out of 32 patients, of aggressive tumors disappeared completely before surgery. Wow. And 66% of the most aggressive triple negative tumors disappeared. Wow. In 12 out of 18.
Now, what are we talking about disappeared? Are we talking about got tired of the treatment or passed away?
Or passed away. Oh my gosh.
Yeah.
In the group with lowest levels of the marker, almost 50% of aggressive tumor. Well, it's talking about the tumors.
Oh, maybe. Oh, you're talking about the tumors.
The tumors disappeared.
Okay.
So that's good.
All right.
Yeah. That's excellent. This was certainly not the biomarker we expected to provide the answer, and that might be why the riddle hasn't been solved before, end quote, says Christopher Godina, first author of the study. The result was surprising. Previously, it was thought that tumors which contained a lot of this particular protein, IGFBP7, could be treated with the medicine. The reverse turned out to be true. The drug is effective for those whose tumors contained a little of the protein. The study, recently published in the scientific journal MPJ Precision Oncology, shows that it is possible to identify the group of patients who will be helped by the treatment. That's awesome.
Yeah. All right. Another quote. A quarter of all patients with aggressive breast cancer may benefit from the treatment. The discovery is potentially something of a breakthrough after several unsuccessful attempts to show which patients benefit from this treatment, says Helena Jernstrom, professor at Lund University and one of the researchers behind the study. It is also possible that patients with types of cancer other than breast cancer may be helped by the medicine, another positive. The researchers say that this result demonstrates the value of open data. Again, if the American researchers had not made their results available, it would have been difficult to solve the mystery. Again, a quote, in this case, it was possible for others to analyze the results and see if they could find something that the earlier researchers had missed. So it all stems from their openness. We hope that this will not now lead the drug companies that have shelved this medicine for cancer treatment to resume its development, concludes Christopher Godina. So, absolutely.
It's like you're looking at the trees and you can't see the forest, and somebody else takes a fresh look and discovers something.
Right. Go to one camp, go to one summer camp, and I'll be there at the same time when you're doing this stuff. So, that way everybody's doing it and they're sharing information. And I don't know. I guess in my naive way, I just thought that is what was going on. Just like when you think about national security organizations and investigative and intelligence organizations, you would think
there's big disconnects.
Yeah, you would think that they'd all be.
You would think
sharing, have a board meeting a month or whatever.
Like we said, Scott, if we create an intelligence cabinet position, then all that's going to get shared. You know what I mean? In one cabinet secretary.
Yep. Well, it's probably what's going to be scary is what we find out that wasn't shared. That's going to be the scary part. So looking forward to that.
Yeah.
Anyway.
All right, folks, thanks for joining us today. Tomorrow we've got, actually, this is the morning segment. This afternoon we have an Egyptology. I like Egyptology. I'm fascinated by it as a kid. I've got a tattoo it's a tribal tattoo of Egypt. So I love it. So you might find it dry, Scott. You might find it dry, but I like it.
I'm in denial, man.
Shut up, man. Okay. Thanks for joining us, everyone. We'll talk to you later.